Welcome to Chapter 4, Section 6, Archaea. Learning Objectives. Describe the unique features of each category of Archaea. Explain why Archaea might not be associated with human microbiomes or pathology. Give common examples of Archaea commonly associated with unique environmental habitats. Like organisms in the domain bacteria, organisms of the domain Archaea are all unicellular organisms. However, Archaea differ structurally from bacteria in several significant ways, as discussed in the unique characteristics of prokaryotic cells. To summarize, the archaeal cell membrane is composed of ether linkages with branched isoprene chains, as opposed to the bacterial cell membrane, which has ester linkages with unbranched fatty acids. Archaeal cell walls lack peptidoglycan but contain a structurally similar substance called pseudopeptidoglycan or pseudomurine. The genomes of Archaea are larger and more complex than those of bacteria. Domain Archaea is as diverse as domain bacteria, and its representatives can be found in any habitat. Some Archaea are mesophiles, and many are extremophiles, preferring extremely hot or cold, extreme salinity, and other conditions that are hostile to most other forms of life on Earth. Their metabolism is adapted to the harsh environments, and they can perform methanogenesis, for example, which bacteria and eukaryotes cannot. The size and complexity of the archaeal genome makes it difficult to classify. Most taxonomists agree that within the Archaea there are currently 5 major phyla: the crenarchaeota, Archaeota, Prokaryotic, Nanoarchaeota, and Thermoproteus. There are likely many other archaeal groups that have not yet been systematically studied and classified. With few exceptions, Archaea are not present in the human microbiota, and none are currently known to be associated with infectious disease in humans, animals, plants, or even microorganisms. However, many play important roles in the environment and may thus have an indirect impact on human health. Crenarchaeota. Crenarchaeota is a class of Archaea that is extremely diverse, containing genera and species that differ vastly in their morphology and requirements for growth. All crenarchaeota are aquatic organisms, and they are thought to be the most abundant microorganisms in the oceans. Most, but not all, crenarchaeota are hyperthermophiles. Some of them, notably the genus Pyrolobus, are able to grow at temperatures up to 113 degrees Celsius. Archaea of the genus Sulfolobus are thermophiles that prefer temperatures around 70 to 80 degrees Celsius, and acidophiles that prefer a pH of 2 to 3. Sulfolobus can live in aerobic or anaerobic environments. In the presence of oxygen, Sulfolobus species use metabolic processes similar to those of heterotrophs. In anaerobic environments, they oxidize sulfur to produce sulfuric acid, which is stored in granules. Sulfolobus species are used in biotechnology for the production of thermostable and acid-resistant proteins called ephidins. Ephidins can bind and neutralize various antigens, molecules found in toxins or infectious agents that provoke an immune response from the body. Another genus, Thermoproteus, is represented by strictly anaerobic organisms with an optimal growth temperature of 85 degrees Celsius. They have flagella and therefore are motile. Thermoproteus has a cellular membrane in which lipids form a monolayer rather than a bilayer, which is typical for Archaea. Its metabolism is autotrophic. To synthesize ATP, Thermoproteus species reduce sulfur or molecular hydrogen and use carbon dioxide or carbon monoxide as a source of carbon. Thermoproteus is thought to be the deepest branching genus of Archaea, and thus is a living example of some of our planet's earliest forms of life. Archaeota. The phylum Archaeota includes several distinct classes. Species in the classes Methanobacteria, Methanococci, and Methanomicrobia represent archaea that can be generally described as methanogens. Methanogens are unique in that they can reduce carbon dioxide in the presence of hydrogen, producing methane. They can live in the most extreme environments and can reproduce at temperatures varying from below freezing to boiling. Methanogens have been found in hot springs as well as deep under ice in Greenland. Some scientists have even hypothesized that methanogens may inhabit the planet Mars because of the mixture of gases produced by methanogens resemble the makeup of the Martian atmosphere. Methanogens are thought to contribute to the formation of anoxic sediments by producing hydrogen sulfide, making marsh gas. They also produce gases in ruminants and humans. Some genera of methanogens, notably Methanosarcina, can grow and produce methane in the presence of oxygen, although the vast majority are strict anaerobes. The class Halobacteria, which was named before scientists recognized the distinction between archaea and bacteria, include halophilic, salt-loving archaea. Halobacteria require a very high concentration of sodium chloride in their aquatic environment. They require concentrations close to saturation at 36%. Such environments include the Dead Sea, as well as some salty lakes in Antarctica and South Central Asia. One remarkable feature of these organisms is that they perform photosynthesis using the protein bacteriorhodopsin, which gives them and their bodies of water they inhabit a beautiful purple color. Notable species of Halobacterium include Halobacterium salinarum, which may be the oldest living organism on Earth. Scientists have isolated its DNA from fossils that are 250 million years old. Another species, Halopharax volcani, shows a very sophisticated system of ion exchange, which enables it to balance the concentration of salts at high temperatures. Microconnections, finding a link between archaea and disease. Archaea are not known to cause any disease in humans, animals, plants, bacteria, or in other archaea. Although this makes sense for the extremophiles, not all archaea live in extreme environments. Many genera and species of archaea are mesophiles, so they can live in human and animal microbiomes, although they rarely do. As we have learned, some methanogens exist in the human gastrointestinal tract, yet we have no reliable evidence pointing to any archaean as the causative agent of any human disease. Still, scientists have attempted to find links between human disease and archaea. For example, in 2004, Lepp and Company presented evidence that an archaean called Methanobrevibacter oralis inhabits the gums of patients with periodontal disease. The authors suggested that the activity of these methanogens causes the disease. However, it was subsequently shown that there was no causal relationship between M. oralis and periodontitis. It seems more likely that periodontal disease causes an enlargement of anaerobic regions in the mouth that are subsequently populated by M. oralis. There remains no good answer as to why archaea do not seem to be pathogenic, but scientists continue to speculate and hope to find an answer. That brings us to the end of Chapter 4, Prokaryotic Diversity. Next time, I guess I'll go through Chapter 5. I'm just going to delete that part and call it good. Okay, bye.